Hello, and a very warm welcome to today's Cell and Gene Therapy Insights webinar, titled Efficient Reprogramming of Fibroblasts into iPSCs using mRNA Technology. I'm Charlotte Barker, an editor at BioInsights, and joining me today are Dr. Parivash Nouri, Hannah Johannsen, and Dr. Sebastian Dieke, who will explore an mRNA-based reprogramming workflow that streamlines the process of creating high-quality iPSCs. After the presentation by Parivash and Hannah, we'll have a live Q&A session with our speakers, and Sebastian as well is going to join us. And I'll just briefly introduce our presenters. Dr. Parivash Nouri is the Application Product Manager for pluripotent stem cells in research within the Global Marketing Department at Miltenyi Biotec. She obtained her PhD from the Technical University of Munich, in which she conducted research on iPSCs and their differentiation into midbrain dopaminergic neurons. Hannah Johannsen is the current group lead in the R&D department with a focus on iPSC technologies and cell sorting, also at Miltenyi Biotec. She's been with the team for over 10 years and completed her master's at the University of Duisburg-Essen. Dr. Sebastian Dieke is the head of pluripotent stem cell platform at the Max Delbruck Center in Berlin. His research involves reprogramming, differentiation, and genome editing of human-induced pluripotent stem cells. So, without further ado, I'll hand over to Parivash to kick us off with the first presentation.
Thank you, Charlotte, for your introduction. First and foremost, I extend a warm welcome to each and every one of you. Thank you for joining us in today's webinar. We will kick off the session with a quick intro to Miltenyi Biotec and exploring the key benefits of our workflows. Following that, my colleague Hannah will guide us through an in-depth presentation of our cutting-edge reprogramming workflow. We will wrap up with a compelling take-home message and a special highlight. Dr. Sebastian Dieke, a valued member of our scientific community, will share first-hand experiences with our workflow, and finally, we will open the floor for a Q&A session to address any queries you may have. Miltenyi Biotec was established over 30 years ago in Germany. Currently, we employ more than 4,500 individuals with over 20% dedicated to research and development. This figure highlights our strong commitment to delivering innovative solutions to researchers and clinicians. With our headquarters located in Germany, we have a global presence in over 70 countries. We operate both through direct sales and partnerships with distributors. At Miltenyi Biotec, we offer a vast product range of over 19,000 items. Today, we will focus on our regenerative medicine portfolio, which covers stem cells and applications in cardiovascular and neuroscience research. Miltenyi Biotec excels with a comprehensive product lineup and advanced technologies tailored to your needs. We are providing not just products, but solutions that ensure reproducibility, minimize experimental variability, integrate seamlessly with our compatible reagents and instruments, and offer user-friendly workflows. With these principles at the core of our service, we are fully equipped to meet your needs for your whole workflow. To support you optimally during the whole pluripotent stem cell workflow, Miltenyi Biotec provides instruments and reagents for all the key steps involved in PSC research, starting by sample preparation and cell sorting over a vast selection of cell culture reagents to flow cytometry and imaging. I will now pass the floor to my colleague Hannah, who led the team to develop our StemMACS iPSC reprogramming kit. Join us as we take a deeper dive into the details of our reprogramming workflow.
Thank you for the introduction, Pari, and I'm happy to take over and guide you on the next part of our webinar through our reprogramming workflow, all the way from the start of the fibroblast isolation to the characterization of your generated pluripotent stem cells. As you all know, iPSCs promise an enormous potential that's represented by the many possible applications of these cells. They are, for example, a powerful tool for studying human disease and development and open the possibility of modeling disease-specific phenotypes. Moreover, pluripotent stem cells represent a virtually endless source of material for drug and toxicity screening. And finally, pluripotent stem cells are prime candidates for the applications in regenerative medicine, where lineage-specific progenitor cells can be used to replace degenerative or diseased cells and tissues. And all these applications are showing why we are in need for efficient methods to generate the cell type. As you're well aware, the generation of induced pluripotent stem cells from somatic cells by reprogramming via the forced expression of several transcription factors is a very complicated process that involves several key events along the way. And one very important step is the complete epigenetic remodeling of the cell and changes in metabolic pathways when the cell undergoes mesenchymal to epithelial transition. And to obtain a fully reprogrammed cell, it needs to be independent from exogenous transcription factors. And how fast this is achieved is actually highly dependent on the used reprogramming method. So over the last decade, we've come a long way, starting with integrative DNA-based reprogramming technologies that were then soon complemented by several non-integrative DNA-based technologies. Unfortunately, those still harbor a risk of spontaneous integration into the genome. In the last years, however, Sendai virus and RNA-based reprogramming were considered the safest as well as most efficient methods for generation of iPSCs. And for our newly developed reprogramming kit, we follow an mRNA-based approach, since mRNAs don't persist in the cells, and therefore no other method guarantees such a fast independence from exogenous factors without the need for screening or subcloning. And that means that the iPSCs can be checked for the quality in very early passage and then used for downstream applications without worrying about any experimental bias by presence of remaining exogenous factors. And in the end, this proves to be also very cost effective and allows, for example, core facilities to generate less clones that can be tested very quickly after generation for subsequent distribution. In addition, reprogramming with mRNAs is very easy from a technical perspective and also allows to work on a low biosafety level. So after this short introduction, I would like to guide you through our workflow for the generation of monoclonal iPSC lines in under 4 weeks that we performed for 5 healthy donors. So we started by isolating fibroblasts from human skin punch biopsies, as you can see here, and expanded them for only 1 passage before seeding them directly for reprogramming. So the reprogramming itself takes around 14 days, depicted here, during which you basically seed the cells, transfecting with our mRNA reprogramming cocktail twice a day for 5 consecutive days. And then allow the emergent iPS colonies to grow a few more days, during which you just have to form some mediate changes. As you can see here below, you will also always obtain a mixture of reprogrammed and non-reprogrammed cells. So a lot of people then pick these colony manually. Manually, we actually isolated the induced pluripotent stem cells directly after reprogramming via MACS technology or with our tidal cell sorter. And to obtain clonal cell lines, we then performed a limiting dilution and seeded the iPSCs as single cells directly after sorting. And this could, of course, also be done with the help of an automated single cell seeding device. The cells were then expanded for a few passages and characterized as soon as possible. So we normally have our first frozen and characterized stock in passage 4 or 5 after reprogramming already. And we're now going to have a closer look at the separate steps of the workflow, starting with the fibroblast isolation. So in our workflow, we used our whole skin dissociation kit in combination with our StemMACS dissociator. And this protocol combines enzymatic and mechanical dissociation and allowed us to obtain single skin cells from human skin punches after just 24 hours. We stained the fibroblasts with CD90, and as you can see here on the left, we have usually around 40% of fibroblasts in the isolated skin cells, with the exception of donor 1 that had a lower fraction. In the middle graph here, you can see that due to a varying number of total cells in the samples, we also obtained varying numbers of total CD90 positive fibroblasts from the different donors. This number is, of course, also highly dependent on the sample quality, the transport time of the sample, etc. But as you can appreciate, even for donor 1, we obtained around 2,000 fibroblasts per square millimeter, which means that for a 6 millimeter punch, we had 55,000 cells. And this is more than enough to use for reprogramming after one passage of expansion, since we usually seed 15 to 30,000 cells per 6 well. And on the right here, you can see that after a short expansion of 5 to 7 days, we lost most of our non-fibrous cell types that were also present in the skin, and basically obtained a pure population of CD90 positive fibroblasts to seed directly for reprogramming. For the short one-passage expansion of the fibroblasts, we used our MSC expansion medium that's xeno-free and doesn't require use of an additional coding matrix. And here on the lower part, you can see some exemplary pictures of the fibroblast shortly after the StemMACS isolation. The next step in our workflow and the core part here is the reprogramming of the fibroblasts into induced pluripotent stem cells. So for this, we used our recently released mRNA reprogramming kit that contains basically everything you need to generate induced pluripotent stem cells from human fibroblasts. It consists of a dedicated reprogramming medium, the StemMACS ReproBrew, a ready-to-use mRNA cocktail, as well as transfection reagent and buffers. One kit is enough to reprogram up to 9 6 wells, and the kit is xeno-free and it works without the need for feeder cells, conditioned medium, or B18R protein. Here you can see the reprogramming process itself a bit more in detail. So we usually seed this fibroblast on Friday and then do the transfections from Monday to Friday to minimize hands-on time on the weekend. As I already explained, you transfect the cells twice a day for 5 consecutive days, once in the morning and then again after an incubation time of a minimum of 6 hours in the afternoon. This is important to ensure constant high levels of the exogenous mRNAs in the cells. The transfections are done via a lipid-based transfection reagent, so you basically just mix your RNAs with the transfection reagent, incubate for 20 minutes, and add it to the cells. After the 5 days of transfections, you perform daily media changes until you see your iPSC colonies emerge, and this should be the case on, usually the case on day 11, so 2 weeks after seeding the fibroblasts. So let's have a look at the mRNA cocktail we use for reprogramming. It is a ready-to-use cocktail that contains 6 transcription factors, coding for the classic Yamanaka factors, plus NANOG and Lin28. And as you might know, no matter how well you design your RNA, cells have professional systems to recognize alien RNA, and transfection of mRNAs into mammalian cells usually results in severe cytotoxicity due to the activation of interferon signal cascades that then induce apoptosis. We therefore added an additional mRNA in the mix, coding for SOX1, which stands for suppressor of cytokine signaling. So SOX1 suppresses this autocrine death loop and enables repetitive transfection and therefore reprogramming. And this mechanism is so effective that no B18R protein or additional immunoevasive mRNAs are necessary to be added to this cocktail. So to get a better understanding of how the reprogramming looks like, we recorded a video of a reprogramming experiment over the 14 days that you can see here on the left. You can see here that the cells change their morphology from a fibroblast spindle-like structure to more epithelial rounded morphology. And you can see here on the left that during the course of this 14 days here an iPS colony is formed on the left part. You can see that a nice clear distinct colony arises in this time. And we also performed, as shown here on the right, a cyclic immunofluorescence with our Maxima platform, where we stained an reprogramming experiment with a panel of pluripotency-related markers, fibroblast-associated markers, differentiation-associated markers. And as you can see here, on day 11 of reprogramming, you can see in the middle this clearly distinct colony that stains positive for a variety of pluripotency associated markers, and you can see that it's can clearly be distinguished from the surrounding non-reprogrammed cells that stain for fibroblasts or differentiation-associated markers. Next, I want to talk a bit about the efficiency of our reprogramming kit. So here on the left, you can see some exemplary pictures of generated iPS colonies on day 11 of reprogramming. And on the left part of this graph, here I depicted the reprogramming efficiency that is calculated by the amount of obtained colonies per seeded fibroblasts. And during development, we performed also many experiments with BJs, and we saw that young fibroblasts of early passage perform comparably with, as you can see here, reprogramming efficiencies of around 4%, but with single experiments reaching up to even a 12% reprogramming efficiency. So this is true for BJs and early passage fibroblasts, let's say fibroblasts below passage 5. And on the lower part, you can see that for higher passage fibroblasts, the reprogramming efficiency decreases. And to understand a bit better what that means, I also depicted here on the right the colonies per square centimeter. So here you can see that even difficult lines or samples where you will obtain maybe only 1 colony per square centimeters, and this would still be 10 colonies in a 6 well, which is more than enough colonies to isolate. And so far, we have also not seen any failed donors where we could not generate colonies at all. And here on the right, you can see depicted an overview of a reprogrammed 6 well here that was stained for OCT3 positive iPSC colonies. And here you can see really the abundance of iPS colonies obtained on day 11 after reprogramming. So you might ask if we made sure that we will not retain any exogenous mRNAs in the iPSCs in this reprogramming system. So let's have a look at this. We performed qPCR over the course of reprogramming and checked for the prevalence of the exogenous reprogramming factors. And as you can see on the right, we have a peak at day 4, which is expected after several transfections. And the amount of exogenous RNAs in the cells then rapidly decreases. We calculated that the value at day 11 corresponds to around 10 RNA molecules per cell. And as you can appreciate in the resulting induced pluripotent stem cells, no exogenous RNA was detectable anymore. So after we now successfully generated our iPSC colonies, let's see how we can isolate them. As I said before, a lot of people choose to pick their iPS colonies manually. We here used our magnetic activated cell sorting technology to isolate TAR160 labeled induced pluripotent stem cells after reprogramming. And you can see here the results shown via flow cytometry. So starting with 40% of TAR160 positive induced pluripotent stem cells after reprogramming, we were able to enrich this to a purity of over 90%. And here you can also see that a few days after max isolation, and please keep in mind that this is also the days after reprogramming, the cells show already a typical iPS morphology. For our 5 healthy donors, we used the tidal cell sorter that enables high-speed multi-parameter flow sorting in the safety of a fully closed cartridge, meaning under sterile conditions. And here you can see some examples, exemplary plots via flow cytometry, where we sorted after reprogramming on TAR160 SSEF4 positive cells. You can see that we started with around 9% of iPS cells after reprogramming, and here you can see that we were able to enrich this to a very pure iPS population of a purity of 95%. And here on right, you can see the data for our 5 donors. You can see that we have variant TAR160 expression after reprogramming. And you can see that no matter the start frequency, we were able to generate very high iPS purities for all 5 donors with over 80%. So after we now isolated our iPS cells and have them as a bulk, what we did next is we wanted to generate monoclonal cell lines, and so we actually seeded the induced pluripotent stem cells after the sorting as single cells. So for the expansion of our monoclonal pluripotent stem cell lines, we used our StemMACS PSC Pro XF medium. It's a xeno-free maintenance and expansion medium for pluripotent stem cells that was optimized for every other day and weekend-free feeding schedules. And in combination with the StemMACS PSC support supplement, it also enables you to use this medium in applications that are rather challenging for cells like single-cell seeding or low-density seedings. So as you can see here, for the clonal expansion of iPS cells, we started by seeding a single iPS cell in a 96 well plates via manual limiting dilution. You can see here an example of a colony that grew in this 96 well, and we're then doing an upscale on a 24 well plate, and from there into a 6 well plate, and you can see here that we have very nice morphologies, and this is made possible by addition of the StemMACS PSC support supplement that improves the performance of the PSCs under challenging culture conditions as the low cell seeding densities. For those interested into a GMP and upscaling perspective, we also offer a iPS Pro GMP medium in combination with GMP cytokines, and we also offer a platform, the CliniMax Prodigy. Here it's a automated and fully closed system. We released a process called the adherent cell culture process, and with that you can basically expand pluripotent stem cells in GMP compliant closed system. So after we successfully generated our monoclonal iPS lines after reprogramming, as I show you on the last slides, the last step now is of course to also characterize our cells. So next we checked expression of pluripotency related markers, and here in this graph you can see the expression of several pluripotency related markers as we have 4, 5, 160, SOX and OCT, and we also checked one differentiation associated marker, CD15. So this is actually a multicolor flow panel that is also described on our website. And here you can see the results for the 5 donors for several cell lines per donor. And you can see here that we have very high levels of all pluripotency associated markers in all lines, and by maintaining very low expression of differentiation associated markers CD15 in all lines of all donors. Here in the lower part, as I said, you can see some exemplary plots of this the flow cytometry analysis, and if you want to know how to do that, you can check out this protocol on our homepage. So next, we of course also wanted to check the differentiation potential into the tree lineage endoectomesoderm of our pluripotent stem cells to really confirm that they are indeed pluripotent. And for that, we used our StemMACS tree lineage differentiation kit. It's a kit that contains different media that allows for 2D directed differentiation into the tree lineages. The protocol takes exactly 7 days, so you seed the cells and have the results for all 3 lineages after 7 days. It's very useful to do side-by-side comparison also of different cell lines. You can then do the analysis either via immunofluorescence or flow cytometry. In case of flow cytometry, we again have here a protocol online that you can check out. And on the next slide here, you can see the data for our 5 donors. And here depicted the differentiation efficiency for the three lineages: endoderm, ectoderm, mesoderm. On the right, you have some exemplary plots. So endoderm is characterized by a double positive expression of SOX17 and CXCR4, ectoderm by expression of SOX2 and PAX6, and mesoderm is described via expression of CD144 positive endothelial cells or CD140B positive vascular smooth muscle cells. And here again, you can see that for all 5 donors for various cell lines, so this is 3 cell lines per donor, you can see that we have very nice differentiation efficiency into all 3 lineages for all donors for all cell lines, confirming their pluripotent state. We, of course, also checked genomic integrity of our generated cell lines. So we performed an assay to check for copy number variation, and here we used the ICS digital test from STEM Genomics. This is based on digital PCR and captures over 92% of recurrent defects in pluripotent stem cells. And here you can see the results for 5 lines from each of the 5 donors. You can see that in the majority we don't have any copy number variations, except for 2 lines, one from donor 1 and 1 line from donor 5. So these 2 lines we would then not consider for any downstream applications and probably discard them. So to look at our lines a little bit more in detail, we started doing whole exome sequencing. We started here with 1 line from each donor, with donor 5 still ongoing. So we do a whole exome sequencing that we can then use as a basis for various readouts. So for example, we're looking at genes as part of an oncogene panel that covers 486 cancer-associated genes, and look for mutations or uncommon variations in there. We also have a list of genes that comprise a pluripotent stem cell panel that we want to check based on the whole exome data. This is still ongoing, and of course we always have the option in future to check also for certain disease-related panels depending on the planned downstream application. And for all this, of course, you can always use the data from whole exome sequencing. So apart from the tree lineage assay, where we checked in general the pluripotency of the cells, we also wanted to see if the cells from our donors are able to differentiate into a more definite fate. So here we tested differentiation into midbrain dopaminergic neurons. And here on the left, you can see the FOXA2 OTX2 expression for 1 line from each of the first 3 donors, and you can see that all 3 lines nicely differentiated into dopaminergic neurons, characterized here by OTX and FOXA2. We also, of course, checked other markers, and we also confirmed this via qPCR. So here you can also see FOXA2 OTX expression for these lines, down here you can also see expression of relevant dopaminergic neuron markers as confirmed via immunofluorescence. Of course, there are basically countless different differentiations that you could do based on the generated IPS lines, and for that we offer, for example, our StemMACS diff-based medium, which is an optimized base medium for differentiation of pluripotent stem cells that can be supplemented with a large variety of cytokines on small molecules, and then that combination, this media, can then be used for virtually every type of directed differentiation protocol in a 2D or also in a 3D approach. And with that, I'm at the end of my part of this webinar, and happy to give over to Pari again, who will not leave you without a take-home message today.
Thank you, Hannah, for the insightful introduction to our workflow. Summing up our discussion, As a comprehensive workflow provider, we manage the entire spectrum from human fibroblast collection to iPSC isolation, characterization, and maintenance. The StemMACS iPSC mRNA reprogramming kit employs an mRNA-based non-viral method tailored for precision on human dermal fibroblasts. Its efficiency is notably high, achieving a 4% reprogramming rate with BJ fibroblasts, and it accelerates iPSC line generation in just 14 days. The kit is an all-inclusive reagent set, including the mRNA cocktail, transfection reagent, and reprogramming medium. In essence, we are offering a workflow with a streamlined process and integrated reagents and instruments to ensure efficiency in iPSC generation. Your attention to this webinar is truly appreciated. Thank you.
Brilliant, thank you very much. Great presentation. I'd now like to invite our speakers to turn on your cameras and join me for the Q&A. Okay, so first question that I had here, I want to ask you, Sebastian, how would you describe your overall experience of using the kit?
Yeah, hello everyone. For the beginning, we used already a couple of years ago the first kits which came on the market, mRNA reprogramming, which were a little bit tedious. You had a 20+ day transfection, and in the end, either it worked nicely or it didn't work so nicely. So either you had a high reprogramming efficiency or not. Nothing happened. So for us, we tested it on 4 different samples so far. Also not young fibroblasts. It was from a patient study, so to say, where we have 500 plus samples, and we picked 4 randomly chosen samples in the age group of 40 to 60 and reprogrammed them. And overall, it worked for all of the 4 lines we tested, which wasn't the case in the previous kits we tested, as I mentioned. And we got around 20 plus colonies we could isolate. And so far we are quite happy with that. In the future, we would like to test other cells like keratinocytes to reprogram them on or using this kit. But this is a little bit more challenging because you have to use different media compositions, and this needs a little bit more adaptation, I would think.
Brilliant, thank you very much, Sebastian. I can see we're getting lots of great questions in. Do keep those coming. Any that we don't get to today, we'll try to reply by email afterwards, so you will get an answer either way. But yeah, do keep those coming. Another question for you, Sebastian. What applications did you explore with the reprogramming kit? You already touched on that a bit, but we'll go into a bit more detail.
So as I said, we only tested adult fibroblasts isolated from a skin sample, and we didn't test other cell types. I think most of the audience are always keen to hear that finally someone developed a kit reprogramming PBMCs with mRNA. We didn't test it yet, but I think also Miltenyi didn't test it yet. So that's why I would say our experience are based on fibroblasts isolated from adult people.
Brilliant, thank you very much, Sebastian. Hannah, can you provide guidance on whether the kit is compatible with different matrices for the reprogramming process?
Yeah, thanks for the question. So we have tested some matrices during development, so it works fine on matrigel and GLTRAX, also on laminin 521 full-length protein, and it also works very nicely on iMatrix 511.
Brilliant, so you have tested quite a range there. And Hannah, another one for you. In the context of the reprogramming stage, what plate format would you recommend for optimal results?
So what we have seen during development is actually that the smaller your format gets, the harder it sometimes gets a little bit to distribute the cells evenly, which is also very critical for optimal results. So we are doing a lot of times a 12 well, 24 well, 6 well. Smaller might get a bit tricky.
Excellent. And another one for Hannah. How long does each transfection step take within the reprogramming stage? And are there ways to optimize or streamline that process a bit further?
Yeah, so if you listen in detail, you realize that, so basically it's a standard transfection. So you mix the RNA with the transfection reagent, incubate 20 minutes, and then just add a dropwise to your cells. And you always also do a media change before the transfection. If you want to save time, you can, of course, do this media change, for example, in this 20-minute incubation time. And then in the end, it will just take you a max of 45 minutes in total, I would say.
Brilliant, some good tips there. Pari, what additional materials do I need to purchase in conjunction with the reprogramming kit?
As Hannah also mentioned before, this is an all-inclusive reagent set, meaning that it has everything that you would need for the reprogramming, from the reprogramming media to the transfection reagent to the mRNA cocktail. So basically, you have everything that you need for the reprogramming.
Oh, brilliant. So no additional materials required.
Well, the additional required material is a matrix, of course, but that is something that is up to the researcher for deciding what matrix they're using, and because it's something that is in every laboratory, they may use their own matrix for the culturing the cells. We did not include that in the kit, but basically everything else is provided.
Brilliant. Another one for you, Pari. How much of the process can be automated?
So in the context of the reprogramming, it's basically a pipetting context. So it's mostly pipetting. We are looking into the automation of the whole process, but for the only reprogramming part, basically customers can do that via a pipetting robot that can be applied.
Brilliant. And one, I think for Hannah here, do you notice that after isolation with beads or by flow sorting, that the iPSCs are prone to stress-related detachment, differentiation, or apoptosis?
So also from data that I've shown, we don't really see that. So of course, it also depends on your protocol. So if you use our MAX beads, we have very nice viability afterwards. And as you see, when we use our tidal cell sorter, which is like a very, very gentle sorter, we could see the single cells immediately afterwards. And we've also checked and like unsorted and sorted cells, and we didn't see any differences in the behavior of the cells afterwards. Yeah, so plating efficiencies as single cells were comparable with or without sorting. So, but this of course also depends on what kind of sorter, for example, you use.
Good news. Another one for you, Hannah. Does your reprogramming kit work with other cell types, such as EPCs?
So during development, so it is optimized for reprogramming of fibroblasts. During development, we have also tested urine-derived renal epithelial cells and also mesenchymal stem cells, and we're also looking currently into endothelial progenitor cells.
Brilliant. Lots of questions for you here, Hannah. People were very interested in the data that you presented. Another one here. Did you check or consider the methylation profiles of your reprogrammed iPSCs? And the viewer says, because it was shown that iPSCs, depending on the method of reprogramming, retain an epigenetic profile similar to their tissue of origin, and that might impair differentiation. Is that something that you looked at?
Yeah, so we haven't looked into it. We're considering it. We haven't looked into it so far. I mean, we're using all the cells that we generate in differentiations, not only the one data that I've shown here, but also a lot of other differentiations. And so we don't see any problems with differentiation efficiency or capabilities of the generated cells. But yes, we are aware of this and might look into it in future.
Okay, watch this space on that one. Question here from a viewer who is curious about mRNA toxicity. After transfecting mRNA for 10 times, so twice per day, 5 days, how many fibroblasts survive, and what are the required starting fibroblast numbers? Hannah, again, I think it's for you.
Yes, so as I said, the there is quite a bit of toxicity also during the repeated transfection. It cannot go without that. But we're taking quite some measures so that the cells don't suffer too much. So I cannot really say a number like how many fibroblasts will survive. Required starting fibroblast numbers. So when we do like really this short expansion and reprogramming one passage after isolation, we see 15 to 30,000 in a 6-well. If we have like older passages of higher passages of fibroblasts that maybe have been cryopreserved in between, then we see sometimes up to double amount, 60,000 cells per 6-well, something like that.
Brilliant. And a question here from a viewer: is the specific mRNA formulation top secret, or can we access those?
So I guess I'm going to take that one as well. So we disclose in principle the mRNAs that are present in the kit, but we don't disclose the amount or the ratio of the mRNAs.
Okay, fair enough. Oh, interesting question here. Someone here asking whether you provide internship opportunities for people interested in this area. I guess that's a question that could go to any of you, really. Let's kick us off.
In principle, yes, but depends on the demand. So we're currently taking 4 people in every year, and it's always kind of busy. So, but maybe I can add something because I forgot to mention something. So we changed the protocol a little bit. What we did is that we changed early on from this StemMACS reprogramming brew to the PBS and to the IPS brew. So early on, after day 5 or 6 already, and we also switched the cells into hypoxic, which helped them to survive. And what else did we do? This is a support element. I think in the protocol it's stated that you can keep it in, or you should keep it in. We took it out after 1 day after we plated the cells. And another side note, I think Hannah had the same experience. If we try to monitor, if you want to make nice movies about the reprogramming process, and you have an IncuCyte and you put it into that machine, then the method didn't work for us because I think the temperature change, taking the pictures is kind of critical for the reprogramming outcome. So this was the only time where we didn't succeed to get anything. But when we changed it and took it out of the IncuCyte imaging system, it worked, as I mentioned.
It's a bit photo shy. Brilliant. And are there any internships on offer over at Miltenyi? Pari, I don't know if you want to answer.
So as I am working in the global marketing at the moment, we don't have an internship, but maybe Hannah can guide you better.
Yeah, so if it's about an internship in the research and development, yes, we also offer internships, or also a bachelor and master's thesis, PhD thesis. So feel free to check out the homepage for open positions if you're interested in that.
Brilliant. Yes, absolutely. Do get in touch, viewer. Wonderful. Has anyone used the kit? A viewer asks here, has anyone used the kit for PBMCs and for human amniotic fluid cells?
So for PBMCs, as Sebastian already said, so far there's not really, it's not possible to reprogram blood cells directly with mRNA technology. If you want to use PBMC as a start material, the only option would be to do the endothelial progenitor cells that are outgrown from them. And for amniotic fluid cells, we don't have any, we haven't tested that. And I'm also not aware of any customer that has done that so far.
Okay, excellent. can this kit, well, I guess we just touched on this, but can this kit reprogram fibroblasts into any kind of cells? And does this have any limitations as of now?
So it's intended for generation of induced pluripotent stem cells. So I assume that the question is maybe directed at trans differentiation, so to generate and differentiated cell types directly from fibroblasts. So this is not the intended use. It cannot be used for that.
Okay. And on the subject of subsequent cell differentiation, are all differentiation protocols adapted for attached cells only? Do you have any recommendations for cell dissociation of iPSCs or differentiated cells in order to perform flow cytometry?
Okay. So the differentiation protocols that we currently have are adapted for iPS cells that have been used in 2D. So this diff-based medium can also be used for generation, for example, of embryonic bodies in 3D. So yeah, I don't know, Pari, if you want to add on that.
So the question is whether we do have the recommendation for cell dissociation of iPSCs or differentiated cells. So basically that depends really on the type of the cells that you are working with. For the iPSCs, we do have the passaging solutions that they are specifically designed for passaging the iPSCs, but for the other kind of the cells, it's basically you really have to see what kind of a cells do you mean, like the differentiated cells.
So maybe I can also add on that second part. So we're quite frequently using a triple-like enzyme, which is a recombinant enzyme. Of course, you can also use trypsin EDTA solutions to harvest your cells. So since this is a xeno-free kit, in our eyes, it does not make too much sense if you use xeno-free media and kits to then use a dissociation reagent that has animal component. But for flow cytometry, usually most dissociation reagents, I would say, work well to ensure that you have single cells that can be used to perform flow cytometry.
Brilliant. And another question here. Have you tested hypoxic conditions in reprogramming?
Yeah, so I mean, as probably most people are aware, is that it's published that hypoxic culture conditions can enhance reprogramming efficiency. So the data that I've shown here is all under normoxic conditions. So we made sure that the kit definitely works under normoxic conditions. As I said, it is known that it can enhance performance under to use hypoxic conditions as well.
So maybe I can jump in here. So as I mentioned before, we switch early on to a hypoxic condition. So when the first colonies appear, we switch because we normally maintain our cells under hypoxic conditions. So we don't switch back. We just keep them there based on the fact that what Hannah just mentioned. And yeah, that stabilized for us some clones from certain samples.
Brilliant. And a follow-up question here. At what point do you switch the reprogramming cultures back from hypoxia to normoxic oxygen conditions?
So as I mentioned, we don't switch back. So we keep them only for differentiation. We go to normoxic condition. So.
Okay, brilliant. I think we may have got time for another, maybe one question. Is the kit efficient for, it's a bit specific this one, is the kit efficient for reprogramming marmoset fibroblasts?
Okay, so I think marmoset fibroblasts are monkey fibroblasts.
Yes.
So I cannot say. So we have it is for human reprogramming, obviously. So I cannot say if it would work on monkey. I guess it would have been. Yeah, you would need to test it for yourself, because I think this also depends on how do you culture the cells before, how do they take the medium that has been optimized for human fibroblasts. Yeah, so I'm not saying it cannot be done, but you would have to try.
So maybe some side notes here. We tried orangutan, but not with this kit. We tried another mRNA kit and it didn't work. So it's worthwhile trying it for orangutan, maybe gorilla and chimpanzee. I would say it's most likely to adapt it right away, I would say. But for marmoset or other monkeys where you have to adapt the culture system quite a bit, I think it's a little bit more difficult because you have to understand which media composition you have to use during the process. But there's a publication where they use the StemMACS OOPS, they call it, StemMACS OOPS protocol, where they add cheer and another component during the expansion that helps stabilizing this process.
Brilliant. Wonderful. Well, I think that's probably all we've got time for today. So thank you again, Hannah, Parivash and Sebastian for answering those questions. Any questions that we didn't get to, we'll try to reply to those by email. So if we didn't answer your question today, don't worry. The webinar will be available on demand tomorrow, so look out for an email from us with the link. And I think all that's left then is to thank Hannah, Parivash and Sebastian once more for a great presentation, and thank you to the audience for listening and sending in your questions. We do hope you'll join us again soon.